Hey guys, welcome to our week of virtual classes. I hope that you guys all have a really great week. Just a quick FYI, if you need anything at all this week, there's a discussion board in Canvas that you can post questions and I can answer them for the whole group. Always free to email me with anything you need as well. I will be in town, probably not a lot on campus, however, as I'll be out of commission at a virtual panel for the rest of the week. So I hope you guys all are doing great, had a nice weekend, and let's dive in. We're going to talk today about nutrients at the watershed scale. So just to remind ourselves about what we did last week and kind of really dig back in to the basics of nitrogen cycling. This figure on the right over here, see if I can get my mouse and pointer to work properly. Kind of want to use this laser pointer guy. Perfect. So the biggest thing that's really exciting and makes nitrogen very fun to understand and research, but also challenging, is that it has these incredibly variable oxidation states. And this allows the element to be incredibly reactive. It moves through many different forms. And the two that we most focus on are this one right here in the middle, ammonium, and this one over here, which is kind of lumped together, nitrate and nitrite. But then it also has these other forms of nitrous oxide, dinitrogen gas, which is very stable by most accounts, makes up a lot of our atmosphere's proportion of our atmosphere. So there's a lot of ways in which nitrogen cycles through these, but a couple key things to remember. We have processes that are energy consuming, meaning they require energy for them to happen. So if you look at this top graph here, we see, or the top flow chart, this is photosynthesis. We are taking nitrate and ammonium and turning it into organic matter. We are basically assimilating this nitrogen through photosynthesis and other assimilatory processes and creating organic matter. So granted, photosynthesis is really, carbon dioxide making carbon. However, through that assimilatory process, we need different elements to build the highly specialized components of cellular machinery, so nitrogen being one of those key components. And it comes into the photosynthesis cycle up here at the top. So this is needs energy to make that happen. On the bottom half of this, you can kind of see these other kind of energy yielding reactions, I guess maybe over here on the side. And we're taking this organic nitrogen or organic matter broadly and doing what we might call mineralization. It's not directly called that. We call it bacterial degradation in this particular graph, but essentially we're getting energy out. And in this case, that would be taking something like glucose, turning it into CO2, but we're also mineralizing that organic nitrogen and turning it typically into ammonium. And this can happen under aerobic conditions or anaerobic conditions. And then we talked about a bunch of these other processes and pathways that happen under both aerobic and anaerobic conditions. The things we're going to focus on today, a little more deeper dive into nitrification, denitrification. We're going to probably not talk too much about these other pathways, but we're also going to talk about photosynthesis and its role in kind of regulating elemental ratios, particularly of nitrogen and phosphorus in waters. So with that, let's keep going. We've talked a fair bit over this class, and I know you've seen this graphic already once before, but I want to remind us about why we care about that processes and what that means for this particular movement of nitrate through the environment. So again, as a reminder, these are little particles of organic matter or little mineral clay particles. But what happens in how these things get generated, we have an increased in nitrification in different kinds of places, particularly aerobic soils as the most likely. Also in overlying aerated kind of stream bed sediments or river sediments or even ocean sediments where there's a lot of microbes there, but there's oxygenated water in contact with that sediment layer. So we can have nitrification happening. And then what happens is that nitrification, remember, that's this process. We're taking ammonium, turning it into nitrate. So we're taking the ammonium, kind of that's little sticky purple circles, and turning it into these very mobile nitrate ions that are moved through the soil profile and out towards stream ecosystems. What this means at scales now, so that's the point of this discussion today, is that we see patterns. Okay, I'll just give you the little cartoon version up here. We have this organic N and organic C being mineralized into some forms of ammonium, being denitrified potentially, and we could have losses, but this nitrification is what we see happening in this aerobic area. Nitrate would have to move down into this anoxic zone to be turned into dinitrogen gas and evaded out. So if we get this process happening in these surface soils that are pretty aerated and there's lots of organic carbon to be mineralized, we can see patterns that look like this. So this graph on the left is, and both of them are actually similar, it's discharge, so that's this kind of gray line here, and then this has output from a nitrate sensor. And what's really fantastic about these kinds of technologies is that we can get really fine scale resolution of what that nitrate signal looks like. And so you can see very clearly that we see a little bit of variation over time, and this could be, external factors or even some diurnal signal here. But what is most important is that we see this rise in nitrate that kind of mirrors the rise in the stream flow. And what's happening in this case, we have some rainfall up here at the top. This rainfall is activating all this nitrate that's in the surface soils and getting that mobilized out. So we see a rapid increase, and then as that hydrograph decreases, we still have a fair bit of nitrate elevation in our water. So this is the water quality that's measured at that same location. Now, this is really common in some places, not all, but many cases you can see these kinds of patterns where we have elevated nitrate happening as that nitrification is mobilizing that nitrate, and then the nitrification is happening to create the nitrate, and then the stream flow rises, and that's in response to that rainfall kind of mobilizing nitrate through that shallow subsurface. Another example and similar kind of pattern, looking at carbon. Same kind of idea, but in this case we're seeing dissolved carbon also being moved through the subsurface and into the stream. So we see the same similar kind of behavior. It doesn't often tail quite as quickly because we have a lot of kind of slow leaky nutrients and carbon coming out of these soils. So when we look at one classic example of nitrogen dynamics, forests are one of the first ecosystems that were studied. These examples I'm going to show you are from the HUBBARD BROOK ecosystem study. It's a very classic and incredibly important set of watershed studies that were done to look at water quality, water quantity, particularly water yield, and understanding flooding in response to all kinds of different forest management practices. So let's dig in. This particular site is located in New Hampshire. They bought the land, and studies kind of started in the mid-1950s. The official HUBBARD BROOK ecosystem study, however, started, I believe, in 1960. So it's been a long time, and if you looked back at that figure over here, you can see these watersheds carved out of the hill slope, and these are over here. These little 1 through 6 up here in the headwaters of this kind of northern edge of the watershed. In this case, HUBBARD BROOK kind of flows west to east and connects down here. And there's also, Mirror Lake is also a really kind of famous lake where there's lots of work been done. But what's really interesting is they did really focused on understanding forest land management, like I said, measuring things like water yield, which is the amount of amount of rainfall that's turned into stream flow. So how much kind of total yield would a watershed have? How much does it take in and store both in plants and soil water versus how much is exported out? And there's also a lot of interest in what does all the activities that we might do to a forest and manage it in terms of water quantity, specifically related to flooding, and water quality. Sorry that this picture is a little bit blurry. I kind of put it in pulled it from a website, and I'm giving you these slides, and there's some links to some really great resources from the Hubbard Book Experimental Forests website. They've taken a really great textbook that I've taught from for years and turned it into a really accessible and kind of summarized version online. So I highly recommend you go taking a look. So some of these figures are from there. Some are from screenshots from like photocopies from my book. So bear with me. Okay, so the first thing I want to kind of talk about with this forest and clear cutting is these ideas of what would we expect. So if you look at this figure, this is started in this, like I said, in the mid-50s, and then what they did was initiated a clear cutting in this gray area. So they basically took down all the trees and wanted to see what would happen to a whole bunch of things. And kind of what you might expect, we see a lot of precipitation variance through time, but it doesn't really change from watershed to watershed. So we have two watersheds plotted here, one that was clear cut and one that was left intact. And then down here in this middle one, you see stream flow, the one that had no disturbance, pretty similar yield and stream flow as it turned out from before, not any major differences, but you see an incredibly big jump in stream flow after that clear-cut event at the disturbed site. You also see a loss or a decrease considerably in the disturbed site, a reduction in evapotranspiration. So let's kind of unpack that. First of all, kind of expect that you would see stream flow go up because you have no trees. And so those trees are not taking up any of that water. And so more of it's being lost to the stream flow. You're also having no canopy to kind of intercept that water and kind of catch it on the leaves and inhibit it from actually hitting the water or the soil surface to begin with, but you're also then kind of not having any of that evapotranspiration from the trees as well. So there's a lot of kind of just the structure of the forest causing this stream flow to go up. And also have a fair bit of surface runoff then, as well as potential subsurface flow being increased as well. So the net effect, increased stream flow. Not big surprise there. However, what's really interesting, and looks like I forgot the title, apologize, is what happens to all of the rest of the water quality. And this is where it gets really interesting and fascinating, and lots of kind of cascading effects happening here. So let's kind of start with the nitrogen cycle. So when we have no more plants, we have no more plants taking up that nitrogen that's in the soil. So we have a reduced rate of uptake from those trees, plants, etc. There is some understory and overstory, and we're basically just taking that overstory out. But by and large, that's the majority of the plant biomass. There's also mineralization of all of that soil organic nitrogen is going to be generating more ammonium than that is getting accumulating in the soil. So that is all kind of reduced. So we got less root uptake, and we're having mineralization of this organic N that's accumulating, turning that into nitrate. So we've got a lot of excess ammonium, and that excess ammonium is stimulating autotrophic nitrification. So remember, ammonium to nitrate. That nitrate is readily mobilized. Recall the picture, a little cartoon of the negative nitrogen nitrate being mobilized and not sticky to those soil particles or organic matter, and that is going to be exported to the stream. So if you look over here on the right, we see that clear-cut event, big bump up in nitrate concentrations, really much higher. And these are concentrations that you would expect in agricultural lands, really, really high concentrations. These are 40 to 50 milligrams per liter, which is kind of astounding. We see that in ag fields, but seeing that in a forest is pretty high. The cool thing, and maybe not unexpected really, but kind of challenging to think through and make sure we kind of mitigate as well, is nitrification generates acidity. So if you look down at this little chemical equation here, we've got ammonium plus some oxygen going to nitrite. Don't have the full nitrate piece in here, just this nitrate part. But we are generating 4 hydrogen ions. So that hydrogen ion is excess acidity. So that equation is shown here. Those little hydrogen ions are going to go through ion exchange reactions with any of the other cations that are attached to those negatively charged soil particles and basically do a swap. And so what happens is then you also, not only do you mobilize nitrate, but you mobilize calcium, potassium, and other cations that are held within the soil profile. Why do we care about that? Those are also really important, particularly potassium, for plant growth. So now not only are we losing and we're not being able to store that potassium in the soil, but we're actually getting export of that out as well. So kind of to summarize, what you can see with this graphic is we have this disturbance happening. Well, let's do the dashed line. It's kind of backwards here. So in these graphs on the right, the dashed is the clear cut. In these graphs on the left, dashed is the undisturbed or the not clear cut. So keep that in your mind as best you can. And anyway, this is a little bit longer period of time. So we're getting back here in the late 50s, early 60s. Here's that clear cutting event, and then moving forward in time up until about the mid 80s. And what you can see very clearly is that same rise in nitrate, and then you see it come down over time as the trees recover, and then it comes right back down. So that's kind of showing that recovery time, but it's pretty long. It's about 10 years. And then sulfate is another one which we didn't talk about in great detail, but we can kind of unpack that a little bit as we go. You can imagine nitrate loss, sulfate actually being utilized potentially, and there's some other exchange reactions happening with this as well. But what is really, I think, most obvious and helpful as we see this hydrogen ion increase very much in line with the nitrate increase, and then there's the calcium. And as these both recover, they really closely follow that nitrate signal. So take home is that, we often talk in this class about agricultural disturbance, and that's an important piece, and we'll talk about that quite a lot because it is such a big part of our local economy and the Midwestern and global economies as well. But there's other land management and land use changes that can have big impacts on what we're seeing in our water, seeing in our ecosystems. And this is one small slice of what they found at HUBBARD BROOK. And it's really cool to kind of dig through the other components. So if you are interested, I highly encourage you click on the links in the slide deck and you can go review some of these pieces and kind of get a sense of how much of the different pieces of that ecosystem are connected. So land disturbance, forest disturbance, really important, and there's been some interesting work done in HUBBARD BROOK that's really helping to understand things like forest fires and various kinds of things happening out west right now. So with that, let's pivot over to our next topic. I really wanted to come back to the Redfield ratio that we introduced early on in the semester, and so we'll talk a little bit about that. So when we think about Redfield, let's start by thinking about this downstream perspective. We just got done talking about how we can cause these or observe this kind of loss from a forest disturbance. So that's one way we might see excess nitrogen loss from terrestrial landscapes. We also can see it in many cases from fertilizer application, either to agricultural lands, which is the primary kind of excess nitrogen vehicle, or we can maybe see it from cities or kind of golf courses or all these kind of other more manicured or used lands in our residential spaces. So, but we have excess leaching, and it causes lots of things that are shown in these pictures, eutrophication in places like the Gulf, like the Chesapeake Bay, etc. These coastal zones are driven by too much nitrogen. In these cases, nitrogen is the limiting nutrient for growth. And what Redfield did and others is understanding why is there excess and why is it limiting, and trying to really understand what those ratios might be is really helpful in understanding what our controls can be on the land to help create better water quality outcomes downstream. So we're going to unpack a little bit about, we've already kind of talked about why there's excess nitrogen. Those are land activities typically. Not always, but most of the time is this excess nitrogen is because we are putting in lots of nitrogen either through smokestacks, less so through that, more so through fertilizing crops. And why is it limiting? That's the question we're going to talk about today. So if we think about ecological stoichiometry, big word, but essentially what it means is ratios of nutrients and elements broadly. And I've shown this graph already, but I want to come back to it and kind of unpack how these were created. So in the early part of the 1930s, mid-1930s, Alfred Redfield measured concentrations of nitrate. Remember, nitrate is the one that we really find most in these waters, not ammonium, because of its mobility. Measured concentrations of nitrate, phosphate, as well as oxygen from various places and depths in the Atlantic, Pacific, and Indian Oceans. And this graph shows his data along with a whole host of additional data that have been collected since then. And they showed just incredibly remarkable consistency. Around 20-1, that ratio kind of has later been refined to move upwards to, or move down a little bit to go to 16-1. And that's nitrate, and you can see that over here on the y-axis, nitrate to phosphate. And he also collected more data on carbon, and others have as well, to create that ratio of 106 carbon to 16 nitrogen to 1. And again, this has changed very little as data has been collected over the past 90-ish or so years. So when we think about ecological stoichiometry, we want to think about that these environmental ratios really help us determine what elements might be limiting for either an organism, population, or even a community. And so the ratio in the ocean that we just were seeing is a 16-1. That's in the water. It also is the same ratio, and it's also reflected in the phytoplankton biomass. So there's this homeostasis that exists. So you might call it Redfield's homeostasis or Redfield's ratio, but this ratio is really important. And these feedbacks within the system between the phytoplankton and any other organisms, but mostly the phytoplankton if we're talking about the ocean, and the water in which they live, there's some stability there that kind of buffers perturbations that happen. There is some interesting caveats, however, because some microbes and algae have some really surprising flexibility that in relationship to this nutrient supply. Organisms can vary their nutrient content based on whatever environments they're in such that they can thrive. So one really kind of common and interesting example are cyanobacteria. In culture, they can range from nitrogen to phosphorus of 5-1 upwards to 100-1. There we go. And that just depends on which nutrient is in short supply. And there's interesting theories about this, and a lot of folks kind of attribute that flexibility to the persistence of this particular species or class of organisms that have survived for millennia, it seems. So interesting kind of flexibility that some have, but by and large, most of these organisms and phytoplankton that they see in the ocean have this very consistent ratio in their biomass too. So when we look back to the ratio, what's really interesting is that this complexity isn't really driven by phosphorus. Remember, we talked about that phosphorus is really only available in mineral form, so it's kind of difficult for phosphate availability to kind of drive the directionality of this ratio. So what happens is, back to the very first slide, nitrogen is highly reactive, many oxidation states. So those different forms of nitrogen are important and buffer all of the biogeochemical reactions that happen within the oceans. And so these little graphs that I now showed up here on the right, I think are pretty helpful. So let's start down here at the bottom with this graphic C. So in this case, we have a nitrate deficit. If that happens, like we're living over here, we see parts of the environment that have low, they have nitrate deficits. They have more phosphorus than nitrate than you would if you were in that Redfield ratio. This, sorry, look at my notes. So if we're down in this part over here in nitrate deficit, these different kinds of nitrogen fixing organisms will do the work to bring it back up to this homeostasis kind of condition. They can live in different niches that exist, either depths within an ocean or maybe in different kind of coastal areas that have different kind of excess, availability to this type of environment that they might need. But essentially, at the time, there wasn't really identified when Redfield proposed this that there were even any N-fixing organisms in the ocean identified at all. But they live in areas where there's ample phosphate and low nitrogen and can really kind of push it over in this direction. So the reverse is more often what we are seeing these days. We have excess nitrate. So if we have excess nitrate, denitrification is the process that pushes that in reverse. So that's that nitrate to N2, and it's trying to bring those areas of excess nitrogen back into homeostasis here. Challenge is this one can happen in oxygenated places, right? However, because those little cyanobacteria can live in oxygenated environments, they can kind of create those anaerobic conditions inside different kinds of cellular walls that allow them to live in oxygenated water. However, denitrification has to happen in sediments typically, and it's areas where there's no oxygen. So low oxygen in the bottom waters or even in the sediments themselves. So this is a little bit more restricted in terms of location. So broadly, really interesting kind of ability for historically small perturbations to be balanced and buffered by the nitrogen cycle. Now, in the past, since excess nitrogen is becoming more and more of a challenge, this homeostasis is pretty uncertain. So we've got land use changes that are causing a massive influx of nitrate. We've got climate change that's warming oceans. And this is not only affecting the rate of biological activity, because as we get warmer, these biological processes increase in their rates. So both algal growth as well as other decomposition reactions like denitrification. But ocean warming also affects equilibrium oxygen concentrations. And so under warmer temperatures, higher temperatures, we have lower excess or lower equilibrium oxygen concentrations. So that is going to shift the change in this denitrification. It might actually make there be greater chance for more denitrification to happen, but it's unclear. We also are having ocean acidification because of this excess CO2, and that is changing rates of nitrogen fixation in different parts, maybe different coastal areas or whatever. And the other last bit of the challenge is a lot of these N-fixers can also create toxins, so another suite of challenges that really release some not so great extra pollutants into our waterways. And then when you kind of lump all this together, when Redfield did this work to begin with, it was very much thinking more about this in the context of, broad ocean ecology, but these things are happening at hotspots near where people live. So not only are we creating harmful algal blooms, potentially just maybe normal algal blooms, but also very unsightly and causing lots of water quality challenges, both in terms of like water for drinking, but also water for recreation. But we can be creating water quality problems related to toxins that are then causing health problems. So these are localized at outlets of major rivers, major estuaries, places like the Chesapeake Bay, a lot of smaller estuaries along the coast of the eastern half of our country, big one where the Mississippi River dumps out into the Gulf of Mexico, etc. So these are places where not only are people recreating and that kind of thing, but also important areas for habitat for various kinds of ocean fish and other kinds of organisms. So kind of interesting and challenging to note that these things don't just happen. They don't happen ubiquitously across the ocean. They happen at localized hotspots. And again, those are unfortunately where we are. So thinking about these things in a way of restoration is like, how can we both reduce these excesses? So either excess nitrate or nitrate deficit, or you can maybe think of this as excess phosphorus. And then how do we kind of maybe enhance some of these processes that move us closer towards this line? And I would say this is kind of where we live most of the time. And so how can we enhance denitrification to help us move closer to this line so we have less of that algal bloom occurring? So with that, I'm going to end it, and we'll come back for another discussion of some of these issues related to nitrogen and phosphorus. You're doing a lab on Tuesday related to data that we have for both, not data that we have, data that you will find for Lake Erie and the Chesapeake Bay. So with that, have a really great week, and I will see you soon.